dystrophy, in which we have a very low amount of white adipose tissue. We don't have enough adipocytes to store the lipid that are coming. Right? So these drive several metabolic complications, which one of these metabolic complications is liver insulin resistance steatosis. That will be the topic of my talk today. So we are very interested in understanding, how adipose tissue dysfunction drive this development of insulin resistance in the liver and steatosis, and we are looking mainly at the role of lipid as mediators, also some endocrine molecules that are secreted by adipose tissue as mediator of these phenotypes. So steatosis is a hallmark, or is a feature that we find in several different liver diseases, that they are collectively called as metabolic dysfunction associated liver disease. And inside this group of disease, you can find from a simple steatosis, in which more than 5% of hepatocytes, they do have an increased accumulation of lipids, but you can also find in this group of disease more severe conditions such as steatohepatitis, in which we find in the liver hepatocyte ballooning, also inflammation and the development of fibrosis. And you can also find in this group cirrhosis and hepatocellular carcinoma. So this disease, they are very prevalent. They affect approximately 30% of the people worldwide. And unfortunately, we don't have a good pharmacological strategies to counteract the development of this disease. Okay, so when we think about the liver steatosis, we need to think that the accumulation of lipids in the liver, they depend on the balance between the amount of lipids that go to the liver. These lipids, they can come from the diet, or they can be released from white adipose tissue, and also the amount of lipids they are synthesized de novo in the liver. So when these two processes, they exceed, the rates of fatty acid oxidation in the liver, and also fatty acid secretion as triglycerides associated to VLDL, this leads to lipid accumulation of liver. So we, in our lab, we were trying to understand, how these processes, they are regulated and how they communicate with each other. So to start this, we start a very interesting project. So we produced a mouse model in which we deleted PPAR gamma specifically in adipocytes. So PPAR gamma is a nuclear receptor that regulates adipocyte differentiation and adipocyte survival. So when we delete the PPAR gamma in adipocytes, this leads to a marked reduction in white adipose tissue mass. So the adipocytes, they just die. So this leads to a severe lipodystrophy. So as you can see here, this is a control mice. They do show a normal amount of white adipose tissue. And you see here the lipotrophic mice, you don't see any fat at all. So if you don't have white adipose tissue, these lipids, they go to the liver, they exert lipotoxic effects over there. And you can see here the liver mass is markedly increased in these mice. So we start investigating these mice. I just put here the adiponectin levels just to show that this is a severe lipodystrophy. Adiponectin is adipokine that is produced, it is secreted by adipocytes, and we are not even able to detect adiponectin in the serum of these mice, showing that these mice, they don't have adipocytes at all. So this is a severe lipodystrophy. And very interesting, these mice, even though they don't have white adipose tissue and brown adipose tissue, they do show an increase in body weight. They show an increase in food intake, and they are, as expected, severe insulin resistant and glucose intolerant, as you can see here in this glucose tolerance test. And also we evaluate insulin levels at several moments in the life, in the lives of these mice, and you saw they are hyperinsulinemic all the time. So they do have higher levels of insulin, indicating that they are insulin resistant. And as I have shown you before, they do show a progressive increase in liver mass over the time. As you can see here, here in mice is 6, 12, and 24 weeks old. They have an incremental increase in liver mass. So what we did next was just take a look in the liver of these mice, and as you can see here, we do see a progressive increase in lipid deposition in the liver of these mice. So this is 6-volt lipotrophic mice. just a little bit of fat being stored in the liver. Here, 12 weeks old, we do see a microvesicular steatosis. And here, in 24 weeks old, we do see clear signs of liver disease with hepatocyte ballooning, some markers of leukocyte infiltration and other markers of liver disease. So next, what we did was just to investigate, measure fibrosis in the liver of these mice. So here's the serous red staining, and as you can see here, they do show a progressive increase in fibrosis, and you confirm this by measuring the expression of some markers of fibrosis as collagen 1 and TGF-beta, and also we evaluate inflammation in the liver of these mice, and we found, especially after 12 weeks, a marked increase in some pro-inflammatory cytokines such as TNF, alpha, and interleukin-1 beta. So showing the clear signs of inflammation. So what we did next was just, try to evaluate in a broad way, what is going on in terms of changes in metabolism in the liver of these mice. So what we did was just took the liver of these mice and we ran a proteomic analysis, and we were able to detect changes in a lot of different proteins, but two different groups of proteins call our attention. So the lipotrophic mice, they do show a marked increase in the content of proteins related to de novo lipogenesis or de novo fatty acid synthesis, and also to some proteins related to mitochondrial beta oxidation and respiration. To confirm this proteomic analysis, we measure by Western blot and by metabolic assays, de novo lipogenesis, and as you can see here, the lipotrophic mice show a marked increase in liver content of acetyl-CoA carboxylase, fatty acid synthetase, stearoyl-CoA desaturase, which are enzymes involved in de novo lipogenesis, and this was associated also with an increase in phosphorylated content of AKT, which is an important inducer of de novo lipogenesis in the liver of these mice. So we also run some metabolic assays, and we're using label acetate, and as you can see here, we found a marked increase in acetate incorporation in fatty acids, indicating that de novo lipogenesis is really increasing the liver in these mice. And one interesting thing that we found was the acetate oxidation, which indicates, carbon flux in the 3CA cycle, was also increased in the liver of these mice. Also, we evaluate some parameters related to mitochondrial function, and as you can see here, we found that lipotrophic mice, they show an increase in citrate synthase activity. So citrate synthase is a surrogate marker of mitochondrial mass, so indicating that probably these mice, they do have more mitochondria. And also we found we also isolate the mitochondria of the liver of these mice, and we measure, mitochondrial respiration in the presence of linoyl, a fatty acid, conjugated to carnitine. And as you can see here, the lipotrophic mice, they show higher rates of fatty acid oxidation and mitochondrial respiration in response to this fatty acid. So what I have shown you so far is that in the liver of the lipotrophic mice, we do have what we call activation of a futile metabolic cycle. So what is a futile metabolic cycle? Futile metabolic cycle occurs when you have the activation of an antagonic process, metabolic process, such as de novo fatty acid synthesis, in this case specific, and de novo in fatty acid oxidation. So futile metabolic cycles, although they are denominated as futile, they do have a lot of functions in cells. And one of the functions of futile metabolic function in the cell is to increase the energy expenditure in cell. So what we did next was to investigate whether this activation of this futile cycle was associated with change in energy expenditure in these mice. So as you can see here, here I have, we put these mice on metabolic cage and we measure oxygen consumption over 1 week. And as you can see here in red, lipotrophic mice, they do show a very strong increase, in energy consumption and energy expenditure over the time. So even, at the light or the dark cycle or the the circadian day. So in addition to that, these mice, they do show what we call a metabolic inflexibility. So what we measure here, the respiratory exchange ratio, which indicate to us which is the substrate that is mainly metabolized to produce energy. So if these mice, they show a ratio around 0.7, this indicates that they are mainly metabolized fatty acids. If they do show a ratio around 1, this indicates they are mainly metabolizing carbohydrates. So as you can see here in red, the lipoatrophic mice, at night, they do show a lower rates, lower hair than controls, indicate a higher fatty acid oxidation. But at the light part of the cycle, they do show a higher rare, indicating that in this moment they are, oxidizing more carbohydrates. So this is what we call metabolic inflexibility. So the mice is not able to follow the normal circadian change in rare that we do see in control mice. So one interesting aspect of this, because these mice, they do not have any fat, we're wondering whether this increasing energy expenditure was somehow related to inability of these mice to keep or to maintain their body temperature. So we did measure body temperature in these mice. What we found was a marked increase in body temperature over the time. So this indicates to us that this increasing energy expenditure is not related to any problem in the ability of these mice to maintain body temperature. It's not an issue with insulation in these mice. And also, we measure activity of these mice in this cage. And as you can see here, the lipoatrophic mice show very low levels of activity. They do move less in the cage than control mice. So that increasing energy expenditure is also not related to change in locomotor activity. Okay? So another important function of futile metabolic cycles is to promote the remodeling of some molecules in the body. So as I'm talking about, a change, a futile metabolic cycle between fatty acids, what we did was we performed a lipidomic analysis in the liver of these mice to evaluate how the lipids were responding in these mice. So as you can see here, we found a lot of different alterations, a lot of different modifications in the liver of these mice, but one thing caught our attention, looking at several different lipid species, such as free fatty acids, triacylglycerol, and cholesterol ester, what we found was a marked reduction in the lipid species containing essential fatty acids, alpha-linolenic and linoleic fatty acids. So we also found this same pattern of this depletion of essential fatty acids in some species of phospholipids, such as phosphatidylcholine, phosphatidylethanolamine, phosphatidyinositol, and also phosphatidylglycerol. So as you probably know, essential fatty acids, they are not synthesized in the body. You need to have this essential fatty acids, you need to obtain them from diet. And what is interesting about this is this depletion of essential fatty acids occur even in these mice that they are hyperphagic, they are eating more than their counterparts. So this indicates to us, somehow these mice, they are using these fatty acids, and we still don't know yet how they are using these essential fatty acids, or preferentially using these fatty acids. So what we did next was to measure, these essential fatty acids, they are used to synthesize other fatty acids, such as arachidonic acid, in the case of linoleic acid, and in the omega-3 fatty acids, like EPA, DHA, in the case of alpha-linolenic acid. We measure the content of these possible products, of this fatty acid. We do see a reduction in some of them, and no change in the arachidonic acid, indicating that at least, this depletion of this fatty acid is not related to their conversion in other types of fatty acid in the liver of these mice. And the same applies here for the alpha-linolenic acid. So what I have shown you so far is that, when you have a severe lipodystrophy, this will lead an increase in fatty acid synthesis and oxidation in the liver, so a futile metabolic cycle in the liver, and this is associated with an increase in energy expenditure and also with a depletion of essential fatty acids in the liver. So what we did next, because these lipotrophic mice, they do have a lot of change in their metabolism. So all we did next was to investigate whether these phenotypes, could be reproduced in another model of liver disease. So what we did was we produced a mouse model of liver disease in which we deleted this protein in hepatocyte, which is pitane. Pitane is a lipid phosphatase that inhibits the signaling in the PI3 kinase, PI3K AKT signaling pathway. So when we deleted pitane in hepatocytes, what we have is a constitutive activation of PI3 kinase pathway, as you can see here by the marked increase in liver content or the phosphorylated contents of AKT. We know that this pathway, PI3 kinase AKT pathway, is a strong activator of de novo lipogenesis. So, and as expected, this deletion of pitane drives a marked increase in liver mass, as a progressive increase in liver mass. And what is interesting about this mouse model, that we are able to reproduce the different phases of the liver disease progression. So here at weight 8 weeks, we are able to see a simple steatosis. When we evaluate the liver of these mice at 24, when they are 24 weeks old, we do see fibrosis and inflammation. So it's markers of steatohepatitis. And if you keep these mice up to 48 weeks old, we are able to reproduce the hepatocellular carcinoma in these mice. So what we did was we took mice, 8 weeks old mice, and we evaluate lipid metabolism in the liver of these mice. And as you can see here, we do see signs of steatosis. They do show a marked increase in triacylglycerol content in the liver. And also, as expected, de novo lipogenesis was increasing in the liver of these mice, even in the fast condition, but both at fast and refed condition. And also all the proteins related to de novo lipogenesis, they were even at the mRNA level or the protein level, they were markedly increased in the liver of these mice. So this is the model was working fine, to investigate the impact of de novo lipogenesis in the liver. So what we did next was to investigate whether this increased de novo lipogenesis was associated with change in fatty acid oxidation, and we measure palmitic acid oxidation in the liver and also linoleic acid. We found an increase in both processes in the liver, even in refed conditions. We also found in the liver of these mice, an increase in acetate oxidation, which indicates that the carbon flux in the TCA cycle is also increased in the liver of these mice. And also we found an increase in citrate cytase activity, indicating that also mitochondrial mass is also increased in the liver of these mice. So we are able to reproduce in a different mouse model the same phenotypes that we saw in the lipodystrophic mice. Using a completely different model. We also isolate mitochondria of the liver of these mice, and we measure mitochondrial respiration in the presence of succinate, and we do see some increased respiration, but also in the presence of two different fatty acids, palmitoyl carnitine and also linoyl carnitine, and we did find, an increase in the respiration in the presence of these fatty acids. So we also measure the protein content of some proteins related to fatty acid oxidation, such as TFAN. TFAN is a transcription factor that regulates the expression of several mitochondrial proteins. So their TFAN content was increased in the liver of pitane-KO mice. We also found an increase in the content of CPT1 alpha and also complex 1 and complex 4 from the mitochondrial respiratory chain in the liver of these mice. So we are able, by using a completely different mouse model, we are able to reproduce, this activation of this futile metabolic cycle. So what we did next was, does this also impact energy expenditure? So we measure energy expenditure of these mice, and we also found an increase in oxygen consumption, indicating higher energy expenditure of of PTEN-KO+, and this was not associated to changes in locomotor activity. And interestingly, we also ran a lipidomic analysis in the liver of these mice, and we also found the same phenotypes that we found with the lipotrophic mice, with a depletion of essential fatty acids in several different lipid species, such as triacylglycerol, cholesterol S, phosphatidylethanolamine, phosphatidylinositol, all of them showing, depletion of these essential fatty acids. So this seems to be, important and common features between different models of liver disease. So the conclusions here is pretty much the same, the one that I showed for the lipodystrophic mice, but here, we find this in, in a different mouse model in which white adipose tissue doesn't have anything to do with this. This is only happen in the liver, okay? Well, we do know, from previous studies that mTORC1 and mTORC2, they are very important components of and regulators of lipid metabolism in the liver. mTORC1 is a protein complex that has as catalytic center the kinase mTOR, and this complex they also have this protein called raptor, which is very important for mTORC1 function. And also we do have mTORC2, and also shares that with mTORC1, mTOR as the catalytic center. And here we do have Richter as a very essential protein for the activity of this process. So to investigate the role of these two protein complexes in the phenotypes that I have shown you so far, what we did, we produced mice with double deletion of PTEN and Richter. So this mice shows a deletion of PTEN and mTORC2 deficient. And also, we produce a mouse also with PTEN plus raptor deletion in hepatocytes. So this mice has a double deletion of PTEN plus mTORC1 deficiency in hepatocytes. So we followed these mice over the time. What we saw was that, as you can see here, PTEN increased liver mass. And this increase in liver mass induced by PTEN was completely abolished when we deleted Richter in hepatocytes, but was only partially reduced when we deleted Raptor and mTORC1 in these hepatocytes. We saw similar phenotype when we measure liver triacylglycerol content, and here we also measure cholesterol ester content. We saw a very strong effect of Richter deletion abolishing the effects of PTEN, but Raptor was not able to affect the cholesterol ester content increase induced by PTEN deletion. We also measure de novo lipogenesis in the liver of these mice by measuring the incorporation of acetate in fatty acid. And as you can see here, the PTEN mice show an increase in de novo lipogenesis as expected. The deletion of Richter completely abolished this, and the deletion of Raptor does not affect at all de novo lipogenesis. This increase in de novo lipogenesis induced by PTEN deletion. And a similar phenotype, we saw seeing the content of several proteins involved in this process. So we also investigate fatty acid oxidation in the liver of these mice. And as you can see here, PTEN deletion increased fatty acid oxidation. This is completely abolished by Richter deletion. But when we deleted Raptor deletion exacerbates, fatty acid oxidation in these mice. We also evaluate citrate cytase activity, a similar phenotype, that we saw Richter deletion abolished the increase in PTEN, but Raptor didn't do anything. And when we measure the respiration of isolated mitochondria in the presence of a fatty acid, what we saw was a similar phenotype as the fatty acid oxidation. PTEN increased oxidation as expected, Richter abolished, and Raptor deletion exacerbated this oxidation in the liver of these mice. And we also measure the protein content of proteins involved in this process, such as PPAR alpha and CPT1 alpha, and we saw a similar pattern. PTEN, these proteins, they are increased by PTEN. Richter deletion abolished this increase in their content, and Raptor exacerbated the content of PPAR alpha, but not of CPT1 alpha. So just to conclude, what I haven't shown you so far is that we do have in liver disease an activation of a futile metabolic cycle, which is composed by de novo fatty acid synthesis and beta oxidation. I have shown you so far that the presence of mTORC2 is essential, seem to be essential for the activation of this futile metabolic cycle. And mTORC1, on the other hand, when we deleted mTORC1, we do have, an activation, an exacerbation of beta oxidation, but no change in de novo lipogenesis. So we don't know exactly, how these phenotypes are occurring. We do think that there is a connection between de novo lipogenesis and beta oxidation, and we are trying to investigate with some metabolites or some post-translational modification induced by de novo lipogenesis can affect beta oxidation, creating this connection between these two processes, but we are in process of investigating this right now. So just to finalize, we are lucky enough to be awarded recently with a facility. So we do have, we established in the Institute of Biomedical Science this Rodent Metabolic Phenotype Center. We do have several pieces of equipment to evaluate mice metabolism. And this is for anyone that wants to use, just contact us, just send me an email, and we can set up your experiment. We are able to measure metabolic activity in mice at different ambient temperatures and different light-dark cycles. We are able to measure mice and rats body composition. We are able to run right now experiments with nude and germ-free or immunocompromised mice. We have the infrastructure to run experiments with these mice, and we also are able to measure the caloric content of diet, feces, and other material that you want to evaluate. So if you want to run your experiments with us, just send us an email. I put here our website, and we will be happy to assist you and to help you to run these experiments. So, of course, I need to finish by thanking all the people that did all this work. The work that I showed to you, this was mainly done by Albert Peixoto, that is here, and part of the work was done by Rafael Moreira, that is here. Also, he's present here, watching the presentation, when he did his PhD in my labs. And also, we could not done this without the support, financial support, mainly from FAPESP, and also with the help of our mainly collaborate. So thanks so much for your attention. I'll be happy to take any questions.
Thank you, William. Very nice talk.
Thank you.
So if I can start by asking you, so what do you think is the functional consequence of the futile cycle? Because I keep thinking in my mind, if you have an accumulation of lipids, that the lipogenesis is getting the stronger hand there.
Right. I think this futile cycle, I think first, I think it's a response of the liver trying to deal with this excessive amount of fatty acid that is coming to the liver. I think this is the first thing. But as I mentioned in my presentation, of these futile cycles, they do have different consequences in terms of metabolism. Of course, one thing that is, I think, is new in this, and we are trying to understand why, is this remodeling of the lipids that occurs in the liver. So what we are seeing is not just lipid accumulation, but there's a change in the fatty acid profile of these lipids, and we don't know why this is happening and why this essential fatty acid depletion is occurring, and what is the impact of this in terms of metabolism. Because this fatty acid, they do generate, for example, arachidonic acid, which is a precursor for the synthesis of a lot of prostaglandins. The alpha linolenic acid is a precursor, also a lot of different lipid mediators that we don't know exactly what are their function in the liver yet. So this opens for us a different, a lot of perspective, try to understand what are the impact of this remodeling of the lipids, in terms of cell communication and regulation of inflammation, metabolism, etc. So we are just getting started on that, but I think there's a lot of things to do to understand the real impact of that on liver.
Please.
Can I make two questions? Okay.
Of course.
Very, very nice, William. Congrats. The first question is about your first part of the talk on the PPR gamma knockout model.
Mm-hmm.
And it seems that this animal has a hypermetabolic profile, right? And it's expend more energy and produce more heat.
Mm-hmm.
But as far as I know, when you take out the PPR gamma from fat, in the brown fat will be actually compromised, right? Completely. So
there's no brown fat.
This hypermetabolism is due to a compensatory muscle activity.
This is something that we are looking at. We do think that the futile metabolic cycles, they do help increasing the energy expenditure of these mice, but we were not able yet to pinpoint exactly what is the contribution of the futile metabolic cycle for that. What we are doing in this mouse model is try to inhibit de novo lipogenesis or beta oxidation and see what is the impact of that on the energy expenditure. But the point is, I'm showing one futile metabolic cycle in this mice, but there's we do have evidence there are a lot of more going on. So we did run some gene expression analysis in the muscle of these mice, and we did find some evidence that the calcium, the circa calcium futile cycle is also activated in the muscle of these mice. We saw a strong increase in calpastatin expression and also in some circus. So we need to look at that in detail. In the liver, we do also have some evidence that in addition to this fatty acid, since fatty beta oxidation, there is also an activation of futile cycle associated with gluconeogenesis and glycolysis. Because these mice, they do produce a lot of glucose via gluconeogenesis, but at the same time, they do have higher rates of glucose utilization, and we do believe that also there's a futile cycle there that is helping to increase this energy expenditure. So what we are doing right now is going process after process to try to discriminate, the contribution of each of one of these to this increasing energy expenditure.
Right. The second question is about the lipid profile, that it kinds of you recapitulate the profile in two models, right?
Exactly.
Like you have less polyunsaturated fatty acids.
Exactly.
But mostly the omega-6 ones.
Mm-hmm.
Which is okay because like these diets has way more omega-6 precursors, right?
Mm-hmm.
My point is, if you look at the fatty acid desaturases that are necessary to produce this polyunsaturated fatty acids, like FADs 1, FADs 2, and maybe the expression of these enzymes could be changed in the liver or even in the adipose.
Yeah, we did measure the FADS1 and FADS2, and we saw an increase in their content in the liver and other tissues of these mice. But when we measure their product, they are increased, but not enough to explain the essential fatty acid depletion. So there's some carbons of this fatty acid going in that direction, but they do not respond for all the fatty acid depletion. What we think is going on is that, and we do have some old papers from the 60s and 70s showing that it seems that mitochondria has a preference for beta oxidation of these essential fatty acids. And we do think that is the case. And this, and we did run an experiment we treat these mice with a label essential fatty acid, and we are trying to follow the carbon of this to see if where these carbons they are going. First, how is the rate of the oxidation and where the carbons are going. So this is in the machine right now. So we should have the answer for this in 2 or 3 weeks. Okay.
Very nice talk. Thank
Thank
you.
you, Marilia.
So I have also 2 questions, but I'll put them together. First thing is whole liver that you work with, right? So what about, let's say, Kupffer cells or infiltrating immune cells? What do you think is the role of inflammation in that? Because we found in cancer, for instance, that the liver is steatotic as well. You have a large infiltration of macrophages that will become something like foam cells. They really tends to take up these lipids, right? So that's the first thing. And about the essential fatty acids, would these immune cells contribute to the vanishing of these fatty acids? What is your idea about it?
These are great questions, Marilia. We are trying to investigate them right now. About the Kupffer cells, what we do see in these mouse models is that when they progress in the liver disease, we do see a replacement of the Kupffer cells for monocytes recruited from the circulation. And this change in the profile of these leukocytes seems to be related to the worsening of the disease. We don't know how. In fact, we are trying to investigate this right now. We are trying to deplete these cells, depleting first the Kupffer cells to see, how the disease progress, and also later on try to deplete the monocytes. They are recruited to the city to see how the disease progress. But I do think particularly that the macrophages, especially the Kupffer cells, they do play an important role, especially, in the utilization of these essential fatty acids. Because they are important producers of, these lipid mediators, that they could be using these fatty acids to foment this process. So I have a postdoc now that she's in Germany, Bianca. She's learning how to isolate these cells. When we are able to properly isolate these cells, Kupffer cells, and also other leukocytes in the liver, and we are be able to look specifically what is going on specifically in this cell population.
We do isolate. You don't have to go to Germany, right?
Oh, she's already coming back.
Periportal, perivenous populations and everything. So,
thank you so much, man. I'll write you. I'll write for sure. There's one over there.
Hi, excellent talk, fantastic mouse genetics. I'm wondering if it's worth looking at autophagy, because that's what I work on. I can easily see a mechanistic link and a possible biological consequence. I see that you have reduction in phosphatidylethanol-amine, which is required for formation of autophagic vesicles. So probably that is affected. And of course, lipids are cleared by lipophagy, a specific ones, and that will cause a more accumulation of lipids. So I'm just wondering if you are interested to look at it.
Very, very, very much interested in looking at that. We start doing some experiments with autophagy and lipophagy using some pharmacological inhibitors, but we are not very successful. We use chloroquine to look at those. We use, I don't recall the name of the other, but we didn't see much. But I think it's the tools. We need to find a proper tool to look at the implication of autophagy and lipophagy in these models, and to see, to try to investigate their contribution. I'll be happy to discuss with you if you could help us to set up an experiment.
We can talk.
That would be great. Thank you so much.
Thank you.
Thank you very much, William. We're out of time, so you can ask him at the coffee break. So there will be lots of food, and then everybody will be happy. So we'll be doing lipogenesis, right?
Yeah, that's our time to do lipogenesis, right?
Right.
That's good.
So our second talk today is Professor Julio Cesar Batista Ferreira. He is a professor at the Institute of Biomedical Sciences and the Medical School at the University of Sao Paulo since 2012. He completed his PhD jointly at USP and Stanford University in the US, where he also pursued postdoctoral training. He served as a visiting professor at Stanford from 2019 to 2021, and he is the founder and director of the Spark Brazil program. His research focuses on cellular metabolism. He is an affiliated member of the Brazilian Academy of Sciences, the Sao Paulo Academy of Sciences, and the Global Young Academy. So over to Julio.
Okay, good morning, everyone. First, I would like to thank, yeah, it worked. Yeah, it's fine. Edesio and Sovan for bringing together this amazing scientific program, very outstanding scientists for these 2 days. So I'm very looking forward to see all those talks that are very exciting. So today I was talking about mitochondrial dynamics and I would say hypermetabolism and futile fuel cycle. In our lab, we do mitochondrial biology. So we try to understand how changing mitochondrial function affects the body under different conditions. So this is what we do. We know probably nothing about hypermetabolism, but I'm happy that Luis is here, who will help us. And futile cycle, I'm also happy that William is here, and he just gave the previous talk, which will help me during my talk. And we also don't know anything about cachexia, and I'm happy also that we have many friends here, including Marilia, that knows about that. So I'm showing some preliminary but new findings about how changing mitochondrial biology affects this condition called cachexia. Okay, so first, what is cachexia for those who are not familiar with? I took this from this journal. This is the definition. It's a complicated, first, it's interesting when people write about something about, it's complicated. It means I have no idea what's going on. So it's a complicated metabolic syndrome related to muscle mass loss, fatty mass loss, anorexia, inflammation, insulin resistance, and changing protein turnover. All these highlighted red words are directly related to changing mitochondrial function. So what we try to establish here is that this cachexia, of course, is a multi-organ, multi-system disease, but it should have some overlap with changing mitochondrial function in our body. And this is what we are trying to show you today, at least some of this relation. And this is a picture of a patient, okay? So a cachetic patient, huge loss of muscle mass. And this is a picture of a mouse that doesn't have proper mitochondrial function, specifically in skeletal muscle. So when the mitochondria do not function well in skeletal muscle, this is what we find. This is the phenotype of these animals. There is also a huge loss of mass. I'm just summarizing what we found. There is a fat mass loss, of course, inflammation, insulin resistance, and change in protein turnover. In other words, cachexia is also usually related to, is a secondary related to a primary disease, for example, heart failure or cancer. It's very hard to isolate cachexia from a previous disease. So in this model, we were trying to establish a condition that even though you don't have a primary disease, you can have cachexia by changing mitochondrial function, specifically in skeletal muscle, okay? So what do these animal have that has a huge loss of mass in the skeletal muscle? These animals, they don't have two key proteins, mitofusin 1 and 2. And these proteins are related to changing mitochondrial morphology. So in our, those pictures that we see in the textbook, that mitochondria are very like boring structures like circle. That's not true. Sorry, but that's not true. In real life, mitochondria are very cool organelles. They are moving around, they're dynamically finding each other, and they can fuse and they can divide. So I say there are scissors that can cut one mitochondria in two smaller, and that's important because you can get rid of those bad mitochondria through autophagy. And there are like glues that you can glue, like two small mitochondria in one bigger mitochondria. And these animals that I showed you the picture, they do not have this, I would say, mitochondrial glue. So mitofusin 1 and 2. So they cannot fuse smaller mitochondria. And that's the phenotype that we found in these animals. And this is just if you go inside and look inside the muscle of these animals, this is what happens. So you can see, this is an electron microscopy. It's so we make like thousands of slides, and then we can scan all of them and put together and have this kind of movie. Then we can go through the muscles and see all this, these black dots here are mitochondria. These are the distribution of mitochondria in a skeletal muscle from just like a healthy animal. And this is the distribution of mitochondria in the muscle of that picture that I showed you. You see the much more like dark structures compared to the control. In other words, when you change those proteins that fuse mitochondria, you have the accumulation of these black dots, which are bigger mitochondria but not functional mitochondria. So you decrease the number of mitochondria, you increase the size. But these mitochondria do not produce ATP as they should, okay? So this is what we see, and here you clearly see it's much more connected, the mitochondrial distribution in the muscle of a healthy animal, and in these defective mitofusin animals, you see like this not well connected and very dark and huge mitochondria in the muscle. And just changing this phenotype, this is sufficient to cause muscle loss. So decrease muscle loss, decrease muscle function. And here is when we quantify those, the video. So here you have the size of those mitochondria and the number. So first, when you don't have the glue that can connect two mitochondria make into one, you decrease the number of mitochondria compared to control. And you also increase the size. But you say, oh, but if you don't have the glue, how are you going to increase the size? Because they swell. So they get very swollen mitochondria. So they're like empty, big mitochondria that do not produce ATP. So that's bad because they can release many signaling factors that triggers like cell death. And these are other data that I want to show you that we have in these animals. So here, just to clarify, so we have the control animals, animals that have the double knockout for mitofusin 1 and 2. This is the green. And we have single, but just focus on the orange bars here. First, this is a mass of the different tissues, so different muscles, also the heart. This is the distribution, like here more green, more blue means higher mass, more like white, less. You see a clear change here. So if you don't have those mitofusin in the muscle, you have a huge loss of mass in different type of muscles. Only the soleus muscle does not change mass compared to control. And the heart is also fine. And it should be double mutation. If you have a single one, it does not affect those mass, muscle mass. And here we can measure the area of this muscle or the fibers. And of course, you change the mass, you also change the fiber. So you decrease the muscle cross-sectional area. And here you can see the same thing. This is the size of the fibers in the frequency. So this is the distribution, the black one, of a, let's say, a healthy animal. And when you don't have those glues that connect mitochondria, you have a shift to the left. In other words, you accumulate more smaller fibers in the muscle. But this is the interesting thing. So you have a clear phenotype when you don't have mitochondrial fusion proteins in the muscle. And then we follow up these animals over during the life. So this is the like healthy animals. You see, this is the survival rate. And you see the orange line here. This is the animals that I'm talking about. When they reach like 15 weeks of age, they start dying. And in like 5 weeks time window, they are all dead. So, but why? This is just a problem that they have in the skeletal muscle. And somehow they start dying. And if you overlap with the body mass of these animals, look this, the green one. At this point, you see here an inflection. So we don't see many change in the body mass of these animals compared to the others. But at this here, in the dotted red line, the start that you decrease body weight, the animals start dying as well. So there's something interesting that say, oh, perhaps a local damage now it's propagating to the system, and then you cause like a collapse. That might be related to energy metabolism, but it's causing actually, at the end of the day, the animals are dying. We don't know the reason of that, but we know that in patients, this is something that happens. For example, if you have patients that has cancer, the patients that have cachexia, they die much faster compared to the ones that do not have cachexia. In other words, when you have a muscle defect, you can die faster if you have cancer, or just a muscle defect per se when you start losing weight. But I say, oh, we have decrease in muscle mass, decrease in muscle function, but these animals are losing weight. So what's happening? They are actually increasing their metabolic profile, they are eating less. So then we try to understand why they are actually having this huge decrease in body weight. I say, okay, perhaps you can decrease body weight because you're spending more energy. So we use that fancy machine that William showed you in his last slide, that we can measure, put these animals in a metabolic cage and see how much energy they are expending. And this is the energy expenditure over time. So this is a cumulative, okay? If you see over, like during the days or day and night cycles, you see the blue line, this is a normal animal, like healthy mice. And red one is the mice that don't have mitochondrial fusion in the muscle. They don't have they have less muscle. They have less functional mitochondria, but somehow they are consuming more energy. They are expending more energy by weight. And this is related to increased oxygen consumption, the VO2. So that might explain why these animals are losing weight at that point, and they are dying at the same time point. So perhaps they are dying because they're spending too much energy, or they are shifting, the pathways that they need to activate to maintain the homeostasis during this condition. So if you're spending more energy, you might consume more oxygen, or somehow you have to keep the balance. And this data is quite interesting. So when you measure just the respiratory function of these animals, just like here you can see the respiratory frequency. So how much the frequency of the breathe in, breathe out. So this is the healthy animal under the baseline. This is what happens to the animals that don't have mitofusin in the muscle. You see the spike here. You have many more spikes compared to the health. In other words, these animals, if you look here, they increase it by like 100 times the frequency of respiration compared to control. So they're respiring, the frequency is much higher, and that might be a reason for that. So we have a theory. We think that these animals actually are trying to compensate the lack of oxygen in the muscle. So that's why they're increasing the frequency. It's similar to like diabetes. Diabetes, you have a huge amount of glucose in the bloodstream, but the muscle is actually starving. We think that here we have the same condition. This phenotype, it's trying to just elevate the amount of oxygen in the bloodstream, but the muscle cannot uptake this oxygen because the mitochondria are not working well. And then you have a huge change in this metabolic profile. But this, I'm not showing any data related to this, but only this. And actually, when induced hypoxia, this is what we see. And then increase the frequency of respiration in the wild-type animal. And then if you don't have the mitochondrial function well in the muscle, you don't see a change. In other words, these animals have a hypoxia-like phenotype. Okay, so they are all during the whole life like lacking of oxygen, so they need to breathe like more and more and more, but this is not sustainable. And then if you explore the data that the metabolic cage offers to you, this is something that we also analyze. So the respiratory quotient, so the ratio between VCO2 and VO2. And as William mentioned to you before, there is a change here during the night and day. So during the night, during the day, the animals use more lipids, and during the night, when they are active, they use more glucose. So the blue one is actually the behavior of a normal animal. And when you don't have the mitofusin in the muscle, this is what happens. It doesn't matter if it's day or night, you are always close to we say the quotient close to 0.7, 0.8. In other words, you are always using lipid as a source for ATP production. Okay, so you lose this flexibility that you should have during the day and night to maintain your, for example, locomotory activity. And here you can calculate what the distribution of this frequency actually of RERs during this period. And you can see there is a normal distribution, the blue one, which is a healthy animal. But when you don't have mitofusins, there is a shift to the left. In other words, these animals actually spend more energy coming from lipids and not glucose. Okay, let's see what happens to the lipid profile. So this is, if you look to the abdominal adipose tissue, this is a wild-type animal, and this is animal that do not have the mitofusin in the muscle. You see here all this white tissue, they disappear here. So that's why these animals are losing weight, and they have that shift in the RER. They are burning fat to produce energy, okay? But it's not related. So you say, oh, but they are burning fat. They have higher metabolism. Perhaps they are just walking more. They are running more, and that's why they have this change. But that's not the case. If you measure their running capacity over time, You see, this is a healthy animal, this is the sick animal. They lose the running capacity, so they're running less. Okay? And they are losing weight, and they are eating more. So if you look at the food intake, they are eating more compared to wild type. So they are somehow trying to compensate this hypermetabolism. But this seems not to be sufficient to keep the balance again. And if you measure, for example, the weight of this white adipose tissue, you see, it's much less. And actually, it's like a reddish. And Luis told me that might be an inflammatory process is going on in these animals. And also the brown adipose tissue. So in other words, these animals are under huge stress, metabolic stress to keep them alive. So okay, if they are eating more, they are burning more fat, let's give them more fat. Let's give them a McDonald's diet like every day and see if they get healthier, okay? Because then you give more energy source. And if it's the issue, they will have a better phenotype. But that's not the case. So when you feed these animals with high-fat diet, okay, it might have some positive effects. So this is when you give the high-fat diet to a wild-type animal. So now you see a huge amount of fat in the belly compared to the animal that was fed with normal diet. And this is when you give the high-fat diet to these poor animals. They do not accumulate fat in the abdomen, in this region. They are burning fat like crazy. And if you take the liver of these animals and you measure the fat deposition, this is what happens. So this picture comes from this animal here. Huge accumulation of liver in the fat. This picture here comes from these animals. Even though they are eating Big Macs every day, they do not accumulate fat in the liver. So that might be good for your liver. Okay, but liver is just part of our body. If you measure the survival rate of these animals, as I mentioned before, just animals under shock condition, they start dying at some point. If you give them a high-fat diet, you see a shift to the left, so they die earlier compared to the animals that do not have a high-fat diet. In other words, giving high-fat diet to these animals is not a good option. They will die faster, even though they have a better liver, phenotype. Oh, just short. We also did the opposite. Okay, if you give too much food, that's bad. What about if you give less food? We know caloric restriction is a strategy that people try to use to increase health span and also lifespan. We did the opposite. So now let's give these animals less food. So we did the same experimental procedure, but now we are giving only 60% of nutrients, glucose and fat to these animals. And amazingly, when you give less food to them, these animals have a better phenotype. So it's completely contradictory, but now you know that's because they activate autophagy. So now they can get rid of those damaged structures inside the cell and make them ATP. They don't need to use as much food as the other ones. Somehow they just control the balance by using internal source of energy instead of using those coming from the food. So okay, these animals, they lose weight, they die. If you give more energy, they don't live better. They have change in mitochondrial morphology in the muscle, but why they have this systemic phenotype? So we went back to those electron microscopy images. So let's see what's going on. And then we tried to go deeper in those structures, and this is what we found. You see this one? I love Big Mac. That's what I'm saying all the time. Doesn't look like a cheeseburger here. You have a bun, and then in the middle you have like the burger and then cheese. This we call the triad. This is what we have in the skeletal muscle. I'll show you here. So this is a skeletal muscle cross-section area. So you have all the sarcomere here. Here you have two nice mitochondria. And here you have the Big Mac. Bun, and then you have the burger. These are the sarcoplasmic reticulum, this circle. In the middle, you have the T-tubule. This is how those cells get the signal from outside to contract. The signal comes from the T-tubule, and then goes to the sarcoplasmic reticulum, and there's a huge release of calcium into the cytosol. And that's why all these sarcomeres here contract when the cell depolarizes. Okay? And this is a picture of a healthy animal, skeletal muscle. Look the picture of the muscle of those poor animals that don't have mitochondrial fusion. Do you see like a cheeseburger here, or here, or here? You don't see anything. What do you see? A huge mitochondria. But you don't see that, that organization, the disposition that we need to have a proper function for this muscle. So probably having this huge dysfunctional mitochondria might affect the main function of the muscle, which is contract, by changing the calcium handling. So let's see what's going on. That might explain why these animals are so bad in health, because they are not, muscle is not working well. So let's see if the muscles actually is not contracting well. So we did two very simple experiments. This is we take the animal and then we measure the grip strength, and just focus on the orange bar here compared to the dark one. So these are the animals that I mentioned to you. So they are weaker compared to control. So the muscle, it's weaker, so, but they have much less muscle. That's why they are weaker. Okay, what about if you take this muscle, we isolate this muscle, we hang in a machine, and then we give shock, and then we can measure the contraction of the muscle, and then you can normalize by the weight of the muscle. Even though it's shorter, you can normalize and see what's the function, the force that it can generate. And this is what we see. You see the orange line here. This is the muscle from the sick animals compared to the healthy. And then you can increase the frequency of the shock. The muscle usually contracts faster and stronger. But these animals, the muscle is very bad. It does not contract at all. So it's not responding. And we know that the calcium is important for this response. Probably the Big Mac, the cheeseburger, is affecting this response. And then it cannot generate force at all compared to maximal force. It's basically nothing compared to what a wild type or healthy animal can generate. So changing the mitochondrial morphology directly affects this contraction of the muscle. So and then we try to just map what are the players that can affect contraction of the muscle. The major one is we call the neuromuscle junction. It's how the signal comes from the neuron and reach the muscle. And you see in the double knockout mice, this is the normal neuromuscle junction from the healthy animal. In the double knockout mice, you decrease the area of this junction. So you have less area to propagate the signal and activate the muscle. And we measure a bunch of proteins that can make that this junction. And basically, they lose the stoichiometry. So these proteins work together. So if one goes up and the other goes down, you lose the stoichiometry and you lose the function. You don't need to care about the name of these proteins, but just see some of them go up, some of them go down. That's not good to keep the function of this junction. Okay? Okay, when you depolarize the muscle, then the signal goes through the cell, and there are other players that actually can capture the calcium signal, and then the calcium. Oh, so this is the burger. Okay, so you have the T-tubule, and you have the sarcoplasmic reticulum here and here. So the calcium goes inside after the depolarization. This calcium induced the calcium released by the sarcoplasmic reticulum through ryanodine receptor. This is really important to release calcium to the cytosol for the contraction. Look the amount of this receptor we see in the muscle from these poor animals. Much less compared to control. So these animals has less receptor. The calcium should be uptake by a protein that can move the excessive calcium in the cytosol going back to the sarcoplasmic reticulum. These are the circa 1 and 2, sarcoendoplasmic reticulum calcium ATPase. They are reduced. And the calsecrestin, for example, are proteins important to keep it to buffer the calcium in these conditions. So in other words, these animals that have problem in the mitochondrial dynamics, they have a huge defect in the calcium handling in the muscle. And we know that when you have a defective calcium handling, that might be an issue, because in the muscle, the most important thing is to control how much calcium release, how much you uptake. Because if you do not control well, this calcium can go to other compartments, for example, mitochondria. We measure the amount of calcium in the mitochondria in these animals that they are fine. They do not accumulate. But there is something interesting that happens when the calcium handling is not going well. This is a defect that usually seen in some conditions, that the SERCA is the protein that I mentioned to you that can uptake the calcium from the cytosol to the sarcoplasmic reticulum, and then it's stored there. The SERCA can be, there is another protein we call sarcolipin that binds to SERCA. Okay, so when sarcolipin binds to SERCA, the SERCA does not uptake calcium, but it still has the ATPase activity. So for this protein to work, you spend ATP to move calcium through this gradient. Okay, so this is how it happens. You cleave ATP, ADP, phosphate, the calcium goes inside. When you have the sarcolipin, which binds here, you block the uptake, but you still spend energy. And this is what William was talking before about. This is one of those different futile pathways that you spend energy, but you do not uptake the calcium. So you're just wasting energy in this condition. So you increase the energy expenditure. And this is what we see in those animals. Remember, they lose weight, they increase VO2, they lose fat mass. So somehow they are trying to produce more energy to compensate for something. And then we try to understand, considering that they have a huge change in the calcium handling, perhaps that might be an issue that's related to the phenotype of these animals. And of course, when we measure the amount of the tunnel and the lead, you see here, this is the amount of the sarcolipin, which is a lead here. In the healthy animal, you barely see sarcolipin in the tissue. But when you don't have the mitofusions, there's a huge increase in the sarcolipin levels. And you know, there is a ratio, a stoichiometry between the lead and the tunnel. And when you lose this stoichiometry, you might affect the function. And this is what happened. Look, the ratio sarcolipin by a circle 1. That's huge. So you're blocking the function of this enzyme, but this is still able to cleave ATP and perhaps spending more energy. And then, of course, we established in the lab an assay that we can measure the SERCA activity in these tissues. So we can measure the specific and also the non-specific activity by using selective inhibitors for SERCA1. The non-selective activity, we don't see any change when you compare the mice without glue and the healthy animals. But when we measure the selective SERCA activity, this is what happens. So when you do a curve with different concentration of calcium, you increase the activity. Okay, this is what happens to the normal animal. And the mitofusion animal, they respond much better actually compared to control. And when we measure actually the maximal activity, it's also almost double the activity in those sick animals compared to healthy animals. But they do have a huge amount of sarcolipin, so this activity is actually not compensating, it's not putting calcium inside the SR, it's just probably spending more energy for nothing under these conditions. So we think this is still preliminary, but we think that this activation of this futile cycle, it's going on in this muscle because the calcium handling is not working properly. But as William also mentioned, usually, for example, in adipose tissue, the futile cycles are activated to produce heat. So let's see if these animals are actually producing heat. So we did a simple experiment. So we exposed these animals to a cold for 4 hours. And then we just an animal with all the parts in the body. And then we removed the brown adipose tissue and did the same experiments. No change in the thermogenesis. Doesn't matter which animal you compare here. We'd only see a difference when you remove the bat. There's a huge drop in the temperature, body temperature, when you don't have the double mitophins 1 and 2. So we think that this might be related to a communication between the muscle and the bat. But what the take-home message is that the increased SERCA activity in the muscle does not affect the body temperature of these animals. Okay? And when we look to the SERCA2 activity after cold exposure, we lose this difference. In other words, the cold effect in the SERCA2, the SERCA1 activity is independent of the genotype that we have here. Just validating our hypothesis that actually this increased SERCA activity in the muscle is not related to a thermogenic profile. Okay? And I had some solutions for this, but the time is short, so I'll just go to the last slide, if I have, but I don't have time. That I want to thank all the lab members who did all this work, and also our collaborators here that made it possible, and also all the financial support. Yeah. So thank you all for your attention.
Thank you for the excellent talk. I have a very simple question. Do you think it's worth checking, metabolites, the metabolic profiling in your system?
We did check. Actually, we did check the metabolite profile in 5 different organs. So 2 different skeletal muscle, liver, heart, and bloodstream.
Okay.
And there is a signature that looks like the signals, this metabolic profile comes from the muscle. Now we are trying to understand what is the signature and if this signature might control the function of other organs. Right. In other words, now we selected 3 different metabolites that we are validating if they are actually connecting those dots.
Right. I mean, it's common to all the organs.
Yes.
Yeah, that's fantastic.
Yes. And it's enriched in skeletal muscle.
Right. Yeah.
So we think that is signal that's coming from the muscle to outside.
Sure.
Yes.
Okay. And yes.
Very nice talk. Thank you. With the knockout, and William is witness to that, I always ask if you have a control for the knockout, because I wonder if the cardiac mitochondria are not at least, partially affected. So do you have a control for cardiac mitochondria? That's the first question. And the second thing is an offer, because we do have cardiac patients that we work with, samples, including muscle samples. So you could have a look at the reticulum if you want to. In the future, we are working with exosomes now, so we are using more the explants, but we normally do have some spare tissue to offer. Okay?
Oh, yeah, thank you. Related to your second question, it's amazing if we can have access to some human samples, at least to see if actually what we found in these animals are actually relevant to human beings. That will be perfect to do. So, yeah, of course, I will talk to you. And related to your first question, we did many controls. So this is a Cre-LoxP model. So we have only Cre, only LoxP. We checked the mitofusions levels in different tissues, including heart tissue. It's fine. We measure, I didn't tell you, but when we measure mitochondrial function, there's a huge drop in the mitochondrial function in skeletal muscle. We also did the same measurements in the heart and the liver. So the heart seems not to be quite affected. It's actually the opposite. When we do echocardiogram in these animals, the heart is working better, which makes sense, because if you need to pump more oxygen to the body, you need to breathe better and the heart works better. So yes, but this is a very important point when you use transgenic models. We know there is a leakage of this Cre-LoxP system usually. Thank you.
Julio, very, very nice data will show to us. My question is about the adipose tissue. The first image you showed, I don't know, maybe William see too. Your brown adipose tissue and the inguinal adipose tissue, it's likely activated. Do you check that with the UCP1 and another like that? Because maybe your knockout is giving a little of browning on these tissues. Did you see some like that?
If I'm not wrong, we gave some tissues. Luis can answer later to Luis, and he measured some mRNAs for those, right? they're not changing. What we do see an increase in the signal related to thyroid hormone. So those enzymes that are inside the tissue, that there's a huge increase. And we know the hyperthyroidism, it's related to increased energy demand. So this is something that we haven't explored, but we are interested in looking more into, more deeper.
Very nice talk, Julio. Thanks so much. I have two questions. One related, your mouse model shows increased fatty acid oxidation, and where this is occurring, which tissue, and the other question is about anaerobic glycolysis in the muscle of this mice. So if you could comment on that, that would be great. Thank you.
So the fatty acid oxidation, it's not in the skeletal muscle, because when you isolate mitochondria from skeletal muscle and you use fatty acid as a substrate, we don't see any change. It's not doing a better job. And of course, when we measure the anaerobic glycolysis, lactic levels, for example, is just go to the roof. From the beginning, 2 weeks after deletion, and there is a change in muscle fiber, so they have a glycolytic phenotype, pretty established. Yeah. Thank you.
Thank you. We'll break for coffee right now, and we'll resume at 10:30. Thank you.
Just a few announcements now. So we're going to have the coffee break at the 5th floor. We take the elevator or the stairs. So we're really grateful to Revity, the company which has afforded us sponsorship for this meeting. And there will be a Revity exposition in the coffee break area. And there is also an Aquaria exposition at the coffee break area for services related to proteomics mass spectrometry. So thank you and see you there.
Eu vou te falar que a sua apresentação não foi transmitida. Aí só fico sabendo no meio também da apresentação. Deixei a galera na aí, mas você estava aparecendo, dava para te ouvir, mas não dava para ver. Não, tranquilo. É só para te falar. Aí você precisa de alguma ajuda aí?
Nossa, eu vou reconectar para não atrapalhar o próximo.
O point desse aqui, ele...
Eu não mexi no pointer. Ele continua do jeito que estava. Eu não achei, na verdade, no gesto. Será que não tá? Ele falou ali. Bom, a aula... Deixa eu olhar. Ah,